Auzubillahiminash-shaytanir-rajeem. Bismillahir-Rahmanir-Rahim. Assalamu alaikum, dear students. I hope you all will be fine and enjoying good health. I'm Dr. Muhammad Ali Hashmi from University of Education Atak campus, and we are studying the subject of Introduction to Biochemistry. Today's lecture would be about the fluid mosaic model of the membrane structure. In the previous lecture, we were discussing about the significance of lipids in biological membranes, and then we studied about different membrane proteins which are present in the membrane, and after that we also discussed about some lipids and lipid-linked proteins, their percentage, how much they are present in the membranes, how much proteins and lipids and then carbohydrates and other things are present in the membrane. And we also had a quick look at the main structure of the membrane with an emphasis on the lipid bilayer. Now we are going to discuss a bit more about the same topic. So this topic still comes under the umbrella of significance of lipids in biological membranes. So the main concepts of this lecture would be the fluid mosaic model of membrane structure, in which we'll discuss how this model tells you about the structure of the biological membranes. And then we'll study about the experimental verification of the fluid mosaic model. Is it just a theoretical model, or is there an experimental validation for this model as well? So we'll study about different techniques which are used to study the fluid mosaic model experimentally. And then distribution of membrane lipids and proteins in the membranes, how different lipids and proteins are distributed in the membrane. I mean, some of the proteins or lipids, they are distributed towards the inside of the cell membrane, some are towards the outside of the cell membrane. So we'll look into these things as well, how different molecules or lipids, proteins, carbohydrates, they are distributed among the membranes. Let's move to the fluid mosaic model of the membrane structure. The demonstrated fluidity of artificial lipid bilayers suggests that biological membranes have similar properties. So mostly biological membrane, including plasma membrane and membrane of organelles and other things, they should be similar in their properties based on the fluidity of their structure. So this idea was proposed in 1972 by S. Jonathan Singer and Garth Nicholson in their unifying theory of membrane structure, which is known as the fluid mosaic model. So these two scientists, they presented this idea that due to the fluidic nature of the artificial lipid bilayer, most of the membranes, they should have similar properties. So this model, the fluid mosaic model, in original form, it postulates that integral proteins resemble icebergs floating in a two-dimensional lipid sea, and that these proteins freely diffuse laterally in the lipid matrix unless their movements are restricted by associations with other cell components. So let's discuss it point by point. The first thing it says is that integral proteins resemble icebergs. Have you seen that in Arctic regions, if you have seen on television or in some movies, that in the ocean there are giant icebergs floating on the water? So it says that the integral proteins, they just resemble like icebergs floating in a sea. What is that sea? That sea is a two-dimensional lipid sea. mean those two layers of lipids are joined together by forces, which is called lipid bilayer, and in that lipid bilayer see these icebergs like proteins. They are floating in that sea. So and these proteins freely diffuse laterally in the lipid matrix unless their movements are restricted by association with other cell components. So these proteins, they freely diffuse laterally. If you look at the lateral view, they just keep diffusing or moving in those in the lipid bilayer sea unless or until their movement is restricted due to some reasons. Those reasons can be that those proteins are maybe associated with some other molecules like lipids, or they have some specific site to attach on. Otherwise, on all these proteins, the integral proteins, they keep floating in the membrane structure. So their movement is free. You should understand it like that. Yeah, let's come to the structure to have a look at it in a bit more detail. So these are the integral proteins, which you can clearly see if you just depict in or visualize in your mind and consider that this blue portion is an ocean, is a sea of two lipid layers. And you can see that these proteins are like giant icebergs. They are floating in that sea of the lipids, and they can diffuse laterally. I mean, they can move in these directions freely in that sea unless if they are attached to some other portion or some other molecules. They can definitely move freely in this lipid bilayer sea. So that was the basic and the main idea of those two scientists, Jonathan and Garth Nicholson, who presented this model. So let's have a look at it in a bit more detail. So fluorescence, how the fluid mosaic model is experimentally verified. Let's have a look at the experimental verification of this model. So fluorescence photo bleaching recovery measurements indicate that membrane proteins vary in their lateral diffusion rates. So this is an experimental technique called fluorescence photo bleaching recovery measurements. In these measurements, we use fluorescent compounds and then see the membrane proteins where those compounds are attached, and that these measurements, they prove that the diffusion rate, the lateral diffusion rate of the membrane proteins is different for different proteins. Some 30 to 90% of these proteins are freely mobile. So ranging from 30 to 90%, some of the proteins, they are freely mobile. They can move anywhere. They can diffuse very quickly. They diffuse at rates only an order of magnitude or so slower than those of the much smaller lipids. So that they typically take from 10 to 60 minutes to diffuse the 20 micrometer length of a eukaryotic cell. So some of them, they diffuse very slowly at such an order of magnitude, or even slower than that of those smaller lipids. And you can see the 20 micrometer length of a eukaryotic cell is a very small length, but even to cover that, they may take from 10 to 60 minutes. So some of the proteins are moving at a very slow rate, and they just keep constantly moving. They are mobile at all times, but their moving speed is quite slow. It's not like they are moving very quickly and you feel some disturbance in your cells. They are moving at a very slow pace and constantly moving. So some other proteins diffuse more slowly, and some, because of submembrane attachments, are essentially immobile. So this was about the some of the 30 to 90% proteins which move freely Some other proteins, they even move slower than these proteins, and then there are still some proteins which are immobile. Immobile means that they cannot move at all. They have certain attachments to the membrane structure. I mean, they are attached to the lipids, or they are attached to some other components, and they cannot move from their position. They are fixed at a specific spot in the membrane. So the distribution of proteins in membranes may be visualized through electron microscopy using the freeze fracture and freeze etch techniques. These are two techniques through which we can study the structure and confirm the fluid mosaic model of the membrane structure, and we can use these techniques to study the membrane structure under an electron microscope, and then we can confirm that there are proteins that are protruding out of the membrane like this, and like we have seen in the previous figure, this one, and about this lipid bilayer structure and other molecules as well. So what are these techniques? Let's move to those procedures. So the first one, the freeze fracture procedure. In this procedure, which was devised by Daniel Branton, what we do, a membrane specimen is rapidly frozen to near liquid nitrogen temperature. So what is done in this technique? Freeze fracture. Freeze. So first part is the freeze one. We freeze a membrane specimen very rapidly to a very low temperature, that is almost -196 degrees centigrade. Why do we freeze the sample? Just so that it immobilizes the sample, it stops all the movements in that membrane, and and it minimizes the disturbance by the subsequent operation, so that when we study the portion of the membrane under an electron microscope, or we make a sample from it, the movement of proteins do not disturb the study. That is why we freeze the sample and we stop all the movement. So freezing stops all the movements in the cell. Next, the next stage is after freezing. It is the fracture stage. So first is the freeze, and then is the fracture. That is the name specifies. Now we move to the fracture. After the sample is frozen, it is fractured with a cold microtom knife, which often splits the bilayer into monolayers. We'll have a look at it on the next slide. First, I just tell you briefly what is a microtom knife. A microtom, it is from the Greek word mikros, meaning small, and temnin, that means to cut. It is a tool used to cut extremely thin slices of material known as sections. So microtom is a very thin knife that is used to cut very thin slices of materials known as sections. So you can imagine that it is a so thin and sharp knife that it can cut the cellular layers or cell membranes into sections. So it is this tool is very important in science, and microtom's knives are used in microscopy, allowing for the preparation of samples for observation under transmitted light or electron radiation. So with this microtom, we cut sections of different samples which we want to study, and then we study those sections under the electron microscope or other with other techniques. So yeah, here is the microtom knife cutting structure. So you can see that this is a membrane, that same plasma membrane we have seen through the fluid mosaic model, and after freezing it to the nearly liquid nitrogen temperature, it is cut with a microtom knife, and often we get lucky to cut it from the center in half, and this is a schematic representation that if it is cut from half, you can see that the interior of these bilayer is exposed, and you can see the embedded proteins and all those structures in that membrane. So that microtom knife will cut it in the half, and now we'll move to the next procedure to study these structure and to confirm that these lipid bilayers actually do exist. So since the exposed membrane itself would be destroyed by an electron beam, if you put that membrane directly under the electron microscope, the electron beam will definitely destroy it. So what we do to study it, we make its metallic replica. What is metallic replica? That is, we make an exact replication of this membrane using a metal. And how is that made? That is made by coating the membrane with a thin layer of carbon. First, what we do, we coat this portion of the membrane with a thin layer of carbon. So that carbon gets the shape of this membrane, like where the protein is protruding out. It will be we will form a structure of carbon on here, and we will cover this whole portion with the carbon, and then we shadow the carbon by evaporative deposition under high vacuum with platinum. We paste the platinum on that carbon with the help of evaporative deposition. Evaporative deposition is basically a method of thin film deposition where we can put a metal on some targeted surface. So we evaporate the metal in vacuum, and then we spray that metal onto that carbon, a thin layer of carbon which we just put on the this membrane structure, and then we remove the organic matter or the carbon by treating with the acid. And when we remove the carbon, we will have a fine replica of the membrane made up of platinum. So that metallic replica can now be easily examined under electron microscopy, and it will not get destroyed by the electron beam. And in the other procedure, the freeze etch procedure, what we do, the external surface of the membrane adjacent to the cleaved area revealed by freeze fracture may also be visualized by first subliming or etching away at -100 degrees centigrade some of the ice in which it is encased. So you know that the sample was frozen, and we fractured the membrane, and we studied its one side through the making its metallic replica. Its other portion where the ice is frozen, so on outside layer it will be ice because the sample was frozen. So what is done to that side in freeze etch procedure is that first we sublime away the ice. What is sublimation? In sublimation, ice will be converted directly to vapors instead of going through the liquid phase. So we sublime the ice at -100 degrees centigrade, and then you see that the ice that encases a freeze fractured membrane is partially sublimed away as to expose the outer membrane surface for electron microscopy. So when this ice is sublimed away, the outside of the membrane surface, you can have a look here, that will also be revealed, and you can then make its replication and study it under the electron microscope as well. So by using these freeze fracture and freeze etch techniques, you can study both the inner side of the lipid bilayer and the outer surface of the lipid bilayer, and that confirms the fluid mosaic model. So let's have a look at an electron micrograph of a human erythrocyte plasma membrane. So freeze etch electron micrographs of most biological membranes show an inner fracture face that is studded with embedded 50 to 85 angstrom diameter globular particles. So this is the outer surface. So this is fractured area where you can see the inner surface of the human erythrocyte, and you can see small protrudings coming out. So these are all globular particles. They are the proteins which are studied in the plasma membrane. The exposed interior face of the membrane is studied with the numerous globular particles that are integral proteins. So these are all integral proteins. The outer surface of the membrane appears smoother than the inner surface because proteins do not project very far beyond the outer membrane surface. So why is the outer surface much smoother than the inner one? Because proteins do not protrude too much to the outside, and in the inside you can clearly see that the proteins are present in there. These little particles correspond to the membrane proteins, as is demonstrated by their disappearance when the membrane is treated with proteases before its freeze fracture. So how we are sure, or we know that these little protrudings, these are proteins. And we know that because if we treat the membrane with proteases to destroy the proteins, proteases are the enzymes that can destroy proteins, and then we do the freeze fracture procedures, this inner surface will be shown as smoother. So that means that protease has dissolved those proteins. And then this is further corroborated by the observation that the myelin membrane, which has a low protein content, as well as liposomes composed of only lipids, have smooth inner surface fracture faces. So as we studied in the previous lecture, that myelin membrane has only 18% proteins. So if you study that membrane with freeze fracture procedure, it appears to be much smoother from the inside as well. So that also confirms the observation that these little protrudings, they are the protein structures. Outer membrane surfaces also have a relatively smooth appearance because integral proteins tend not to protrude very far beyond them, as we just discussed about it. Now I have inserted two animation videos of the fluid mosaic model of the plasma membrane, just to help you get an imagination, or so that you can visualize in your mind how it would actually look like. So these animations will help you visualize the structure of the plasma membrane and how the movement of proteins occurs in it, and these will also help you to further clarify why the phospholipids are arranged in a bilayer, why there is a bilayer, how they are arranged in it, and what is the function of the embedded proteins in it. So now let's watch these videos and then come back to the topic.
All cells have an outer membrane called a plasma membrane, which acts as a boundary between the outside and inside of a cell. Cell membranes are primarily made of phospholipids, which are large lipid molecules, similar to triglycerides. Like triglycerides, they contain glycerol and fatty acids, but they differ from triglycerides in that they have a phosphate group, rather than a fatty acid, attached to the third carbon of glycerol. The phosphate head of the molecule is hydrophilic, or water-loving. This means that it interacts well with water. The fatty acid tails, however, are hydrophobic, or water-hating, and do not interact well with water. Because of these unique properties, when placed in a liquid environment, phospholipids tend to arrange themselves into a phospholipid bilayer, so that only the hydrophilic heads interact with a watery environment, and the hydrophobic tails crowd inward, away from the water. The plasma membrane of a cell also contains other molecules that aid in its function. There are a number of different proteins embedded in a membrane, and they serve a variety of functions. Some are involved in the transport of substances across the membrane, while others help in cell-to-cell communication. Short chains of sugars are attached to the outer surface of some of these proteins, forming glycoproteins, which serve as recognition signals to other cells. Cholesterol is present in the membrane as well. Cholesterol lends support to the membrane and regulates fluidity, giving the membrane the general consistency of olive oil. The phospholipids, proteins, glycoproteins, and cholesterol are all present in the membrane as a flexible pattern, commonly referred to as the fluid mosaic model. Together, these components of the cell membrane allow the cell to interact with its external environment.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane just 2 molecules thick.
Yeah, I hope you have watched the videos and you now visualize how these phospholipid bilayers arrange themselves and what is their function and how are the mobile proteins moving in their structures. So now we have a look at how membrane lipids and proteins are distributed unevenly. So the distribution of lipids between the different sides of biological membranes has been established through the use of phospholipid hydrolyzing enzyme known as phospholipases. So through use of phospholipases, it is established that how the phospholipids they are present on the outside of the cell and inside of the cells unevenly. Phospholipases cannot pass through membranes, so that only phospholipids on the external surfaces of intact cells are susceptible to their action. So as these enzymes cannot pass through the membrane, so we can just put them on one side of the cell membrane and they will act only on those lipids. So such studies indicate that the lipids in biological membranes, like the proteins, they are asymmetrically distributed between the leaflets of a bilayer. As we studied in the previous lecture, that proteins were distributed in an asymmetric fashion. Similarly, lipids are also distributed in the lipid bilayer in an asymmetric fashion. Carbohydrates are located almost exclusively on the external surface of plasma membrane. So let's have a look. This is the inner leaflet of the plasma membrane and this is the outer leaflet of the plasma membrane. If you look at the phospholipid, that is definitely present on both sides, and if you look at sphingomyelin, that is present more on the outer leaflet than in the inner leaflet. Then phosphatidylcholine is also present more on the outer side. Phosphatidylethanolamine, that is present more on the inner side, and then phosphatidylserine is present only on the inner leaflet of the plasma membrane. So that is this schematic representation shows the distribution of phospholipids in the human erythrocyte membrane, and through this you can understand that the distribution of these lipids is not symmetric, rather it is asymmetric. That was all about the fluid mosaic model and today's lecture, and here is a question for you which you need to find its answer, and it says which intermolecular process primarily derives the formation of a bilayer when phospholipids are added to water. So you have to find that process which is responsible for the arrangement of these phospholipids in a bilayer fashion. So that concludes the lecture, and here are the books that were consulted for the preparation of this lecture. Thank you for watching and your attention, and if you have any question you can post in the classroom and ask your teachers about it, and I wish you the best of luck for your career. We'll see you in the next lecture. Until then, Allah Hafiz.